All right. So I was tasked with the organizing committee here to give somewhat of a primer on low pass. So this is truly going to be a primer. We're going to sort of go back to the very, very basics of what is DNA variation, right? What are reads? What is coverage? Just so that everybody that's here is speaking the same language as we get into some of these more technical talks about construction of reference panels, about how do we utilize this data, about all of the factors that go into successful implementation of low pass in our in sort of a production and industry setting. So again, this is going to be like taking a weightlifting class at 8 a.m. in the morning, followed by like organic chemistry and physics and calculus afterwards. So with that, I'm going to jump right in to the, maybe not. All right, we'll use the clicker here maybe. All right, there we go. So just again, to go back to the very, very basics of all this, right? Is we're talking about all the genetics that we do is quantitative geneticists and animal breeders, right? All of this is dictated, or at least the genetic portion of this is dictated by mutations that occur in the genomes of these animals. So the DNA variation that occurs in livestock populations is going to drive the genetic differences that we're selecting on in breeding programs. The ones that we're most familiar with, of course, this is no stranger to anybody in this audience, but single nucleotide polymorphisms or SNPs, right? Single base pair differences between individual animals. For example, this AC allele on these two chromosomal segments that I have here. The contemporaries, of course, to SNPs would be an insertion or deletion. So a small multi-base pair chunk of DNA that either gets inserted or deleted from a place in the animal's genome. So the important thing to keep in mind about SNPs and indels is as we go out and discover these, this is going to be very population and sampling dependent, right? If we sequence 3 animals, we're going to discover a different number of SNPs than if we go out and sequence 3,000 animals, right? So the more animals that we sequence, the more variation across the genome, genetic differences that we observe, but keeping in mind that this is not linear, okay? And this is going to be really important as we move into this world of thinking about how do we accurately represent all of the haplotypes or as many of the haplotypes as possible that exist in these populations so that we can do accurate imputation downstream, right? There's a plateauing that happens here where we're sequencing more animals, but we've probably observed most of the existing haplotypes that exist in the population. So again, just the very basics of DNA variation. This is the currency with which we work with on SNP chips at a reduced subset, as well as in the world of low-pass sequencing and imputation. So before we jump into sequencing and low-pass basics, I think it's important for us to remember where we're coming from with SNP chips. So SNP chips have been the workhorse for us for the last 15 plus years. Beef, dairy, all livestock industries. And the sort of way that I would lay out how SNP chips have worked is we found fixed locations that we want to genotype, right? These tend to be high minor allele frequency. We want them to be as variable as possible across the population. We need them to be evenly spaced so we can effectively genotype these segments of chromosomes as well as possible with as few markers as possible. These require SNP discovery. So before we can make a SNP chip, we've got to go in and discover the variants that we're going to put on that SNP chip. And so again, there's a lot of legwork that goes into making one of these chips happen. Warren sort of whistled through the F-250 construction, but that was a really monumental challenge of designing probes, finding those functional variants, putting them on a chip. And again, the biggest thing perhaps that we run into with chips is this idea of ascertainment bias. And this is not unique to chips. We'll come back to this as we start to think about imputation in a low-pass context. But the discovery population in which you're observing these initial variants is going to dictate what variants end up being. That sort of high minor allele frequency, evenly spaced variants. There's going to be a level of bias on those variants that get put on a chip due to the discovery population that we're having to use to discover those variants. And the crux of how SNP chip genotyping has been implemented is that it's enough resolution for us to do genomic prediction. As we think about other applications, and we'll discuss that as we dig into the weeds a little bit more here later, but this is a sufficient number of SNPs to accurately represent those chromosomal segments and represent the relationships between individuals, right? And there's been a lot of work that's been done across groups that say that this core set of SNPs, whether it's 20,000, 50,000, 100,000, is more than sufficient to represent the genomic relationships between individuals, right? So as we think about moving into the low-pass world, and again, something that I'll mention at the end of this talk and something that I know will get talked about as we move throughout the day, is we have to be able to put to use the extra information that's contained in a low-pass sequence and imputed genotype. Because as is, if we're just going to use it like we do a chip genotype, right now maybe there's the applications or the advantages maybe aren't quite as clear as if we figure out ways to put that extra variation to work. So let's jump into talking a little bit more about the nuts and bolts of low-pass sequencing. But before we do that, I want to go back and again do a very basic overview of genome sequencing and resequencing and how we parlay that into this context of low-pass sequencing. So all of this is sort of reliant on this idea of shotgun sequencing, right? And shotgun sequencing, we go all the way back to the construction of these first genomes of species. Human Genome Project, of course, a multi-billion-dollar, decades-long project to sequence all of the genetic variation or all of the DNA in a subset of humans, and then construct it back together to have a linear representation of the human genome. And so the sort of approach to all this is when we're starting from scratch without a reference genome, and this is where we started back in the early 2000s as sequencing starts picking up. This is effectively, we're breaking the genome into chunks, right? So readable chunks that we're able to go through and read the sequence of those individual small subsets of DNA that we break out of that animal's 30 pairs of chromosomes in the case of a cow. We read through those small DNA chunks, and then we utilize the overlapping chunks or the overlapping sections of these DNA chunks to construct them into a linear representation of the sequence of an animal, right? This assembly algorithms development has come a long ways from the very beginning of sequencing. But again, the basics of it all, right, is we're basically trying to find sufficient overlap between these chunks of DNA such that we can place them in the correct physical spot. Lots of additional technologies have come onto the scene. I think that Bob might talk a little bit about that. We'll mention more in subsequent talks about the pangenome, but ways that we can utilize sort of long-range connections between DNA, whether that's long-read sequencing, whether that's utilizing physical interactions between chromosomal segments to help scaffold and put together these chunks of DNA. But for our purposes, again, as a primer, this is sort of the most simple and straightforward description of that. And so once we've gone through this assembly process and lots of iterations, again, of manual curation, multiple data types in accurately putting these in the correct physical position related to one another, we have this reference genome that serves as a backbone for future sequencing efforts, right? So we're not, every time we go out and sequence an animal, having to reconstruct this reference genome for every individual animal. Saves us a lot of time and a lot of money and makes things like low-pass sequencing possible. So in our case in the beef cattle industry, this is, of course, Dominette. This is a relatively inbred line 1 Hereford from Fort Keogh. She was sort of hand-selected, again, for as low levels of genetic diversity as they could find in that population, because at the time, Dominette's low levels of genetic diversity, low levels of polymorphism allowed this construction in the form of making a reference sequence to be a more straightforward process because we were dealing with less variation across her genome compared to say, a more conventionally bred Hereford just out in the general population. So after we have a reference genome, this is where our individual genomics comes in, and it becomes, again, a more straightforward process because we're not having to go through this assembly step on all of our genomes that we sequence. right? We're still breaking the genome into smaller chunks, right? We're sequencing these anywhere from 100 to 200 base pairs of DNA. And instead of having to assemble them and figure out their most highly probable relationship to one another in a vacuum, we're able to align them back to this reference genome that we've constructed. So we can take a Brahmin animal, sequence its genome, and align that back to Dominant. It's not going to be a perfect match. There are going to be sections of that animal's genome that maybe are missing in the linear Dominant reference. But for most of our purposes, as we do resequencing of genomes, as we would call this, it's sufficient for us to place those chunks of DNA as accurately as possible. And so, again, no need for de novo assembly, and this allows us to call and identify the differences that drive our genetic differences downstream, right? So we can find places in the genome as we align these back to the Dominant reference genome. We can find SNPs that exist, places in the genome where our animal's DNA appears to be different from the sequence in that linear Dominant reference. And so to keep in mind, this is a picture of an actual reference genome. It's Homo sapiens because I couldn't get a screenshot of my Dominant reference on the server last night. But for now, this is, again, a linear representation, a haploid representation. So you can tell we've only got one sort of line that represents each of these chromosomes, a big long string of A's, C's, T's, and G's. And it's an essential tool for anything that we're utilizing that relies on position, where positioning is important, right? So if we're trying to again, call SNPs at a physical place in the genome, understanding that linear reference is absolutely essential. But it's important to keep in mind, and as we talk about the pangenome later today, this content is based on one animal, and that one animal is an inbred hereford animal in the form of Dominant. There's a lot of genetic variation out there, large chromosomal segments that maybe aren't represented in this Dominant reference. There are alleles that occur even in the hereford population that might not be present in the reference of Dominant. So again, as we move into future chapters of the cattle genomic space, understanding the limitations of a single linear reference genome is something that we have to keep in mind. But for our purposes in this primer on low pass and on sequencing, important to understand that this is really a pretty straightforward text file that just represents the genome of our reference animal in a single string per chromosome. And so we're going to spend the rest of the day today basically talking about low coverage genomic sequencing and how we utilize imputation to add value to it. So I think it's essential that we have a common agreement on what coverage is. Coverage is calculated on a genome-wide basis, not on a site-by-site basis. And the important thing to keep in mind about these sequencing reads is that they're completely random, right? There's going to be a random distribution of these reads across the genome. And so if we're doing, say, 1x, right? So that's the number of reads times the length of our reads. So again, these will vary somewhere between 100 and 200 base pairs long for our short read technologies. And then scaling that by the genome size. If we're out sequencing sequoia or redwood trees whose genomes are like 100 times larger than cattle, it's going to take a lot more reads in order for us to come up with an equivalent coverage. But again, this is a function of how many reads, the length of those reads, and how big is the genome. And again, it's calculated based on the reference genome as we align these reads back to a reference. What is the average depth of coverage genome-wide that we're able to come to when we sequence? So as you can see in this example, there are places in the genome that maybe have, they're covered by 5 reads. There are areas of the genome that are covered by 1 read. And I'll show in subsequent slides here as well that our confidence in calling variants as being real variants and not just sequencing errors is very dependent on the number of reads that we have covering that particular mutation. And so, again, just an example here where as we add more reads at a site, we become more and more confident in calling these variants that might exist at a particular location. So in this case, we have our linear representation of a reference in a focal SNP there, or a focal position there where a T occurs in the reference. We look at read 1, And if we have one read here, and at that same position, when we align the most probable alignment back to the reference genome, we see that there's an A that occurs. With a single read to support that, our confidence that there's a true mutation, a true SNP that occurs there in that particular animal that we're resequencing might be relatively low. But as we read through subsequent reads, right, we see another T pop up. Again, that A, we're maybe less confident that there's a SNP there. It might just be a homozygote. But as we add more supporting reads, at this point, with 4 reads, we might say, all right, that was just a sequencing error in read 1. There's probably not a SNP there. But as we add more reads, we become more confident. We observe that, that A mutation in another subset of those reads, which tells us that this animal is actually likely a true heterozygote at that particular position. But again, with a single read to support that, we're less confident in calling that as a true SNP. So again, our confidence scales with the number of times that we cover a position. The number of times that we cover a position is a function of that average coverage genome-wide. So then moving into low-pass sequencing, the differences between low-pass sequencing and normal sequencing are that you do less sequencing with low-pass, right? There's really not a core difference other than the number of reads that we're utilizing on a single sample. And what we see with low-pass sequencing, and generally as we talk about low-pass, it's anywhere from the low end would be 0.1x, the higher end maybe would be a 1x coverage, so an average coverage of 1 across the genome. But what we see, even with 1x coverage, where we have an average of a single read covering the entirety of the reference genome, is there going to be places where we have multiple reads that cover a single position, where we can pretty highly say something about the variation that might occur at those more higher depth of coverage positions. And there are areas of the genome that don't ever see a single read covered, right? So we're reliant on these areas that get covered by sufficient reads in order to call genotypes or represent genotypes as a dosage with some level of confidence, such that we can use higher coverage animals in imputation, which we'll talk about here in a second, to fill in these missing spots that aren't supported by either a sufficient number of reads or any reads at all. And so the important thing to keep in mind about low pass in that context is that it may not be useful in genomic prediction on its own. So we go out and we sequence this little paragraph, right? And we have a sort of a smattering of text here. It's hard for us to piece together the full importance of what's going on in this little paragraph of text. Imputation is our tool to fill in those missing pieces, right? So we can utilize this information, the positions of variants that we call with some level of confidence in order to fill in the missing stuff. So as you can see here, this is an all-time great song, Rocky Top, that we sing a lot in Tennessee. But again, this is imputation filling in the missing parts that we're not able to observe with that low pass genomic sequencing. We can do the same thing with chip data as well. If this is a chip representation of a bunch of samples, we can use higher coverage animals, whether that's sequence, whether that's a higher density chip, in order to fill in the missing pieces and get a more complete picture of the variation landscape in that individual animal. And so this is what an imputation looks like in practice. This is from a paper that we did a couple years ago here that compares. I pulled out just markers that exist on the SNP50 in this large QTL region of the GWAS that we run. And as you can see, this SNP50, which again is more than sufficient for representing genomic relationships, we can make a genomic relationship matrix that is just as accurate as with full sequence data, with that SNP50 representation of the genome being much more sparse. But as we try and map, dare I say, causal variants or variants that are perfectly associated with causal variants, that becomes much, much easier as we have the resolution from a sequence imputed data set that you'll see down there. a very complex LD structure around this particular location, and allows us to dig in and refine those variants that might be important for downstream genomic predictions or for gene editing, whatever your use of a likely causal variant or haplotype might be. Our ability to map them is much higher with imputation, whereas with the SNP50, we don't see a single one of these variants that reaches our threshold of significance. So imputation, again, I'll go through a really basic rundown of what imputation is doing. This is an example from a really nice paper that was published, I guess almost 13 years ago, that lays out imputation in the context of array-based genotypes. But the same sort of theory in general comes when we start to think about low-pass sequence genotypes as well. So in this case, we might have a subset of data, right? This is a reduced representation, array-based genotype. And as we utilize that directly in a genome-wide association study, we're likely going to be missing some of the variation that might be causal or that might be most significantly associated with our causal variant there. What we do with imputation is we're able to leverage a set of reference haplotypes. And this is something that I know that Bob Schnabel will talk about here in a little bit. But I'll lay out the really basicness of this. What we can do with the individual genotypes down here is what we call phasing. So we can figure out which parent each of those segments or haplotypes of the genotypes that we see in panel A there. We can assign those to parental haplotypes and then utilize information on high coverage or high density reference haplotypes to fill in the missing genotypes that exist on those haplotypes after we've done phasing at the genotype level. So again, there's a lot of algorithmic importance. There's a lot of other information that we can leverage, and Bob will do a really good job, I'm sure, laying out the importance of accurate pedigrees as it comes to phasing and imputation. We can borrow a lot of information that we record in other areas to make sure that we're assigning those haplotypes correctly and imputing the correct genotypes back in at those missing locations in the genome. And the end result here after phasing and imputation versus this reference is a higher density set of genotypes in which we're maybe able to better refine those causal variants or important potentially functional variants when we run an association study. And in the context of genomic prediction, there's possibly some application there, and we'll get to that here later on in the day. And this is just a quick rundown from the Glimpse algorithm. So this is, again, I won't get too far in the weeds here with the algorithms that are used in low-pass sequencing, but the basics of it are really quite similar to what we would see in a chip imputation pipeline. So again, we have areas of the genome here. These are 3 different samples whose genome is covered in a low-pass manner, I would say. This might be 1x coverage or something along those lines. And you'll see that some of these areas, so in the context of this little zoomed in spot here, individual 1, the far right variant, there's no reads that cover that. So there's an equal probability starting out that there's being a 0, which would be a homozygote reference, a heterozygote, or a 2 would be a homozygote alternative allele. So again, with no read support at that location, there's going to be an equal probability of each of those 3 genotypes that occur at that position. And then as you see, as you move over to the next position, 2 reads that support that, or maybe there's 3 reads, I can't quite tell. But again, the more read support we have, the more confident we are in the genotype that exists at that position. So in this case, that would be a homozygote reference genotype at that one. And then as we move down to the next potential SNP, with a single read of support, right, there's maybe there's a heterozygote there, potentially that confidence is a little bit lower. So again, these are all what we would call genotype likelihoods or genotype dosages. This is a measure of our confidence based on that sequence of what the actual genotype might be at that position. And then effectively what happens is this haplotype selection and search, utilizing a probabilistic model to assign the most likely reference haplotype. So again, we're going back to this idea of having high coverage animals. What's the most likely reference haplotype given that information of the genotype likelihoods that we have from the animal's low-pass sequencing data. It calls certain haplotypes at the end. It gives us posterior estimates of that genotype probability. So again, these things are going to be delivered in sort of a confidence level. So the more support that you have, both at the sequencing level and at the level of reference haplotype support, the more confidence this posterior genotype that comes out the other end of a low-pass sequencing algorithm will be. So an important thing for us to keep in mind as we think about choosing these and throwing these into a genomic prediction context is what do we do with these places in the genome where we're not very confident in the imputed genotype, even if it might be one of those core SNPs that's important to how we do evaluation. And so thinking a little bit more about what accurate imputation needs, again, Bob will talk more about reference panel construction, but I'll introduce some of the ideas that we've uncovered in doing high-density chip reference construction and how that we might parlay that into the low-pass world. So we've got to have this large set of reference haplotypes that's representative of the samples that we're trying to impute in. Imputation isn't going to magically dream up a haplotype that it doesn't see in its reference. So in order to do as accurate as possible of imputation, we've got to have a reference that is representative of the population that we're imputing. And again, this idea of high-quality reference genomes, understanding these actual physical positions in recombination is going to play a big part in this as well. But we're going to focus most of our attention here on that large set of reference haplotypes. And imputation's accuracy, whether it's low pass, whether it's at high density chip level, is very dependent on allele frequency, right? We're quite good at imputing high minor allele frequency variants, but when we get into that low tail end, it gets a lot more challenging for us to accurately impute rare variation. And so as we think about the impacts of rare variation on complex traits, understanding that we're going to have to represent more animals in that reference population in order to get that tail, those low minor allele frequency variants, imputed more accurately, it's going to be highly dependent on the animals that end up in that reference population. And one thing that I'll throw out here too is that as we think about the accuracy of imputation, there's going to be a level of error that occurs, right? This is an inevitability. We're going to impute markers incorrectly at certain places. Those tend to be more rare markers, but it's also important to know that Illumina chips aren't 100% accurate either, right? We're not getting 100% accuracy with every genotype that gets called on an Illumina chip. And that's going to be a more randomly distributed errors rather than these errors in imputation where we have a little bit more of an idea about where they're going to occur, which variants we should be more worried about having imputation errors in. I'm going to skip through that so that we can get to the good stuff here. Imputation, again, will only impute what it sees in a reference panel. And so representation really matters when we think about doing accurate imputation at the low pass or even at the chip level. And so the other thing too to keep in mind is that having more animals, more individuals in a reference panel, it's not going to hurt the imputation ever in our experience, right? But not having the right animal in a reference panel is what can damage our imputation when we look at animal. It certainly is. So we've got some good examples here from a paper in 2019 where we compare the impacts of utilizing a breed-specific versus a multi-breed reference and how accurately we're able to impute from low-density chip up to the union set of the F250 that Warren mentioned earlier in the HD, so about 900,000 variants. But we really think that the lessons here likely spill over into what's going to happen in the context of low pass. So admixed populations, so populations that have open herd books in particular, are almost always going to benefit more from having a multi-breed reference than a closed herd book breed would. But even in cases of, we can think about the Angus population, borrowing haplotypes from high coverage, say Sim Angus animals, there's likely going to be a benefit to having those animals that have a chance of ancestry and being able to borrow haplotypes and do more accurate imputation. So in both of these cases, this is the same plot, but just zoomed into rare minor allele frequencies. The green is a breed-specific reference. The Purple is a multi-breed reference. And as you see, particularly as we get down to the low minor allele frequency variance, that separation between the multi-breed and breed-specific references starts to pull out quite a bit more. And again, this is representation of those rare alleles in the reference panel, allowing us to do a better job. We're borrowing more of those from breeds that might not share a breed label with the individual that was being imputed. And also important to keep in mind in these public genomic data sets as well, that an animal's labeled population isn't necessarily the ground truth be all end all, right? Lots of animals that get dumped on the short read archive, which is our big sort of repository of sequence information that say they're one thing, but you look at the DNA and they're very different things, right? So we can still borrow information from some of those animals, particularly when we utilize a multi-breed reference that leverages a large amount of haplotype diversity. But this is where I think this is really nicely illustrated. So this is a small subset of gelding animals that we imputed. The number of imputation errors being on the y-axis there. And you'll see that in the breed reference, there are a good number of animals that are imputed with quite high accuracy, right? These animals have very few imputation errors. And when we look over here at their corresponding imputation in the composite reference, there's a similar number of imputation errors, right? We impute those animals that are well represented in the reference quite well. But the animals that have poor imputation, so that have, 7,500 or more imputation errors in the breed-specific reference, we almost always do a better job imputing them over here in the composite reference, again, because we're borrowing more information from a more diverse set of haplotypes in that multi-breed reference population. And so as we move into thinking more about low pass, I'm going to switch gears and talk just a little bit about the advantages in future areas that I see low pass being an important development for us in the genotyping space, and then compare and contrast a couple of things that are really, really important for us to keep in mind, particularly about the scale of data that exists in low pass and what that's going to look like at the level of a genetic evaluation. So the first and most important thing I think for low pass and the advantage that we have is that the potential for further cost reduction is still there. Our chips are always we're gonna have to synthesize bead pools, right? We're gonna have to continue to make chips. There's a fixed cost associated with those that's harder to chip away at, no pun intended, than what we have in low pass, where there's still a fair bit, I think, of extra cost savings that we can squeeze out over the long term. The ability to represent rare variation, not need to redesign chips as we identify new informative markers. And then again, as Warren talked about earlier, the potential for doing CMV and SNP discovery in low pass is something that we also have the opportunity to do that we wouldn't have in the context of chips. So with that, I want to compare and contrast really quickly here the difference in sample processing, because at the moment, this is probably one of the bigger impediments to us moving into a low pass world. So chip processing a sample to put on a chip is pretty straightforward, right? Amplify DNA, fragment it, precipitate it out, put it on the chip, and visually image the chip, all right? This is a pretty straightforward process. That's very easy to automate in something that I think there's, again, the preparation in order to get that on the chip is quite cheap and quite straightforward to do. Whereas sample preparation for sequencing is much more labor intensive if we're doing it by hand for certain. It almost requires some level of automation, particularly at this third step of library preparation. There's a lot. This is a multi-step, very complex thing that we have to do in order to prepare these chunks of DNA that we've fragmented out and added adapters to. In order to make that sequencing library, there's a level of technical expertise. It's labor intensive if you're doing it by hand in the lab. And again, something technically difficult that maybe doesn't occur necessarily at the level of chips, right? And then beyond that, there's a whole level of data processing and bioinformatics that has to happen on the back end in order for this to become, go from this level of reads to a usable genotype that we can have and utilize in a genetic prediction context. Whereas what comes off of the Illumina sequencer, when we're utilizing a chip genotyping array, this is something that is basically user ready as it comes off of the machine, right? So a level of bioinformatics that has to happen after the fact that's not happening for free and is going to be something that's time and labor intensive. And so the actual data, you'll hear talk today, I'm sure, about FASTQ files. So this is what our deliverable off of a sequencer is going to be, whether we're doing low pass or high coverage genomic sequencing. And what this is effectively every base pair that's sequenced in a large file that also has information about an individual's, the quality at each of those base pairs that we sequence. This is going to be very important for downstream bioinformatics, ensuring that we're putting good information into these algorithms and softwares. Whereas the thing that comes out post-imputation is actually going to be a smaller file, but it is going to have an order of magnitude or two more information than what we would get off of an array-based genotype. This is a variant call format file, or a VCF. This is going to include only our called and imputed SNPs and additional metadata off of those. A smaller file than what's going to come off the sequencer in the form of a FASTQ once we've gone through the processing parts of an imputation and variant calling dialog. And so just to compare and contrast scale, I think this is important as breed associations, genotyping companies think about this shift from chips to low pass sequencing. So for a single animal in a 100K array final report, I looked this up yesterday as I was making slides, 0.003 gigabytes worth of information on an Illumina final report for 100,000 SNPs on a single animal. The same animal with 1x coverage in a FASTQ file is going to be about 3 gigabytes worth of information. So as we think about the long-term storage of this, this is multiple orders of magnitude additional information, but it's also multiple orders of magnitude additional storage that we have to be responsible for at the level of breed associations, genotyping companies, academic groups, and so on. And even process data, as we think about delivering 25,000 animals on a 50K array, again, just what I had handy yesterday as I was flipping through files, about 17.5 megabytes in a BCF format. So this is a compressed variant call format, whereas when we look at the same animals in a similarly compressed variant call format, 30 million variants, we're up well over 10 gigabytes worth of information. So even the processed data, there's still a couple of orders of magnitude more information that we have to handle, store, and figure out what we're going to do and utilize this within a genetic prediction context. And so the big questions surrounding storage, I think, are really about what do we store? And this is a conversation I'm sure that we'll continue to have throughout the day and tomorrow as well. Do we store all of our raw data? These are big files, even compressed. We're talking about terabytes of information, even in fairly small data sets. Do we store just imputed sequence, knowing that this takes time and resources in order to get from that what comes off the sequencer to what we're able to actually utilize in the form of a variant call file? Or do we just store a core set of SNPs? And the important thing to keep in mind, particularly as we think about these last two, if we're not going to store FASTQs long term, what happens as we update our reference? What happens as we get new methods that we're able to better do imputation? These things are not going to be as static as a chip genotype because they're going to be very dependent on what comes in from the side of a reference panel or an imputation software. This length of storage, I think, is something that's also we've started thinking about even at the level of chips with millions of chip genotypes. How long do we need to keep these things around before they become a burden, but don't necessarily add anything from an imputation or a genetic prediction accuracy standpoint in our evaluation? How often do we re-impute? So is this something that we're doing on a weekly basis, similar to genetic evaluation? Do we re-impute just once a year as we make big changes to a reference panel? And then ultimately, the question we'll have to sort of tangle with in the immediate term is, as we make a shift towards low pass, how are we going to integrate this with chip data? There are a number of different ways that I can see that we would do this, but none of them is particularly straightforward. And then the million-dollar question is, how do we use the extra data? In the immediate term, that might just look like using this same as we do SNP chips, right? We get delivered 30 million SNPs from an imputation service, and maybe we just extract our 50,000 SNPs that we used to make a GRM, our 10,000 core SNPs in our genetic evaluation. In this case, right, this allows us to immediately use low pass. It's congruent with our typical genetic evaluation that utilizes chips. We can use these things side by side, but we're not getting any extra value in this case out of utilizing our low pass sequencing and imputation approach for genotyping. The next thing that people have proposed with this scale of data is that if 50,000 SNPs is good, must 30 million be better? There's limited evidence that that's the case. So unless we're actually making changes to the way that we do genomic predictions, this additional variation isn't by default going to improve the quality of what comes out the other side of single-step GBLUP, for example. And then I think the sort of unknowns is if we can accurately prioritize variants, whether that's through some sort of a GWAS and fine mapping, functional annotations and whatnot, might we be able to improve the quality of our genetic predictions with some of this functional biological variation? I think the jury's still out there, but it's going to require that we utilize models that can handle non-normal variant effects, such as Bayes RC and so on. But again, the applicability of those at production scale genomic predictions is something that I think we're still, the jury is very much out on. So with that, I'll leave you with a couple of takeaways, my contact information, and I'm happy to take questions here for the next couple minutes if you've got any. Yeah.
So if you were to skip the RC on storage, where you're just storing in a core set of SNP, is there any functional difference between that and like a genotyping chip per se in terms of the value there that can be once we update asset feature?
Yeah, if you're just storing a set of core imputed SNPs, right? The ability with a new reference panel, a new software, anything that might improve the quality of that imputation, you're going to have to go back to square one if you still care about that genotype being useful in the future. So I think that's where things really diverge from a low pass to chip genotyping standpoint. Storing that core set of SNPs, or rather that core set of SNPs itself, is dependent on things that happen upstream of what's getting stored. That makes sense.
So, Troy, do you have actually, I mean, at some point, what evidence do we have to how much actually changes when you change the reference population? What evidence do we have of how much changes when you change the imputation software program? I mean, I appreciate that you bring it out. The question is, how much is the magnitude of the change?
Yeah, that's going to be really, really population dependent. My guess is that right now we can do a pretty good job of imputing Holstein, and in the future, even if we sequenced 100,000 more Holstein animals at high quality, we've probably got most of those haplotypes represented in a current reference. We're not going to see much wiggle room there. We go to a population like Brahma, for example, where we have limited representation in the reference panel. The addition of core animals, right, core haplotypes being represented could have a pretty big effect on what comes out the other end of that imputation. So it's going to be very population dependent. And as we make those updates, making sure that animals that were imputed on a previous reference are still congruent with animals that are being imputed on an updated reference or with an updated software is going to be something that we've got to have a good handle on. Bob's got an answer.
Sure. I've got a slide about that.
He's got a slide on it.
Okay, so do I make on it after.
And so does Cedric. Good.
Troy, maybe a follow-up question
Sure.
about storage. You're talking about much more like a genomic level. You can do strategies on like animal level, like key animals being stored with raw data, things like that.
Yeah, that's certainly an approach that would be worth considering, right? Those animals that you determine as core animals, or particularly, I think, and Mark might talk about this later this afternoon, core haplotypes that are important that you store. Thinking about animals or thinking about this genotype information at the level of the haplotype rather than of the individual animal and what gets stored, such that you could back, sort of back impute maybe even from that imputed set of markers, core SNPs, whatever you want to call them, that you generated from a larger population of animals. I think there's a lot of different ways to skin the storage cat, and that's certainly one thing to think about is figuring out which animals are absolutely essential to keep around for the long term, knowing that if you're genotyping 100,000 animals a year, a couple gigabyte large fast Q files are going to fill up your storage really, really, really fast.